No? Okay. Then let me ask Devangi to give you a quick revision summary. Devangi, will you do that for Iram, your fellow classmate? I hope you both are friends by now.
Yes, sir.
Yes. Okay, so let me just give me 1 second. Let me put this thing here. For the sake of connectivity, I'm just keeping my video off. I'm on a limited network connection. Is my voice clear? Okay, good. So Devangi, would you like to give a quick summary to Iram about respiration? What did we do?
Yes, sir.
Yes.
So we started with respiration in plants. Plants exchange gases like oxygen and carbon dioxide through their stomatal openings. Then we talk about cellular respiration. It is the mechanism of breakdown of glucose in the presence of oxygen to release energy and use this energy to make ATP. Or we can also say that cellular respiration is the oxidation of respiratory substrates. Then apart from carbohydrates, we have proteins, fats, and some organic acids. They can also act as respiratory substrates. During oxidation in the cell, the total energy present in respiratory substrates is not released freely into the cell in a single step, but instead it is released stepwise in stepwise reactions which are controlled by various enzymes.
Correct.
Then finally, all this energy is trapped as chemical energy in the form of an ATP molecule. And ATP is known as the energy currency of cell. It is utilized in all energy requiring processes within the organism.
Yeah.
Then we have a question why plants have no specialized organs for gaseous exchange. So there are several reasons for plants not having respiratory organs.
Okay.
First point is that every part of the plant takes care of its own gas exchange needs, and there is very little transport to depend on.
Yes,
Then,
little transport of these gases to depend on. So what Devangi means to say, let me just elaborate in one sentence for Iram, is that like in a human body, the cells in our leg, in the calf or in the foot, they cannot take their own gas from outside. So we cannot breathe through our skin. So there is nothing on our skins to breathe from. So our whole body, every organ, liver, kidney, pancreas, all these for oxygen depend on the blood circulatory system. And on behalf of the whole body, lungs do this job of taking in air and giving out the CO2. So we have a centralized organ to do that. In plants, it's different. In plants, there are stomata in every leaf, right? So every leaf, and in fact, every plant, every part of the plant which is exposed to the atmosphere can take gases through diffusion through these small pores present on the surface. Is that clear, Iram? So there's no need for a centralized organ. Okay. Does it make sense? Yeah, continue, Devangi.
so plants do not have greater demands for respiratory gaseous exchange. Only during photosynthesis, large amount of gases are exchanged, but each leaf can take care of its own gaseous needs.
Correct.
And the living cells in the plant are located close to the surface of plant body. As exchange through diffusion is not efficient in plants, and they don't have a concept of body fluid network.
Yeah.
Then we have the chemical reaction of respiration. In respiration, complete combustion of glucose takes place. So we have C6H12O6 plus 6O2 gives 6 carbon dioxide, 6 water, and energy.
Yeah.
Then we started glycolysis. It is the breakdown of glucose to pyruvic acid through an enzymatic process without the help of oxygen. This happens in all living organisms, bacteria to humans.
Yeah.
In
And pay attention to the part that out of this chemical reaction, the overall reaction, we have not started utilizing oxygen yet. So it is without the help of oxygen. So glycolysis can happen in all kinds of organisms, whether they are aerobic or anaerobic, because it does not require oxygen. Yes, continue.
in anaerobic organisms, glycolysis is the only process of respiration.
Oh, yeah, right.
Then scheme of glycolysis was explained by Emden, Meyerhoff, and Parnas. So it is called EMP pathway. Then we started steps of glycolysis.
Yeah, so before you start, before Devangi starts, let me tell Iram that everything which is written in black, okay, is the name of the molecule, and in blue is the energy, whether invested or obtained. Green is the enzyme for that particular step, and in red it is the step. And if it is a single arrow, it is a one unidirectional step, and if it is a this kind of what is it, backflow arrow going in both directions, it is a reversible step. The steps can be back. Yes, Devangi, so I'll do that. So Devangi wants me to go over these steps. So let me go over these steps, okay? So glycolysis, basically as the term suggests, we have to break down glucose. Glyco means glucose, lysis means breakdown. So let's start with glucose, which is a 6-carbon molecule. And the first few steps of glycolysis does not give out any energy, but in fact, they take energy. So you can see that ATP is coming in and getting utilized. So this is called the investment phase, where what happens, you take glucose and with the help of a glucokinase, and kinases are the enzymes which add phosphate on any substrate. So in the first step, you can see that they use energy and convert glucose into glucose 6-phosphate, which is a 6-carbon molecule. It stays a 6-carbon molecule. No breakdown has started yet. So all these steps where energy is either invested or energy is produced becomes a irreversible step. So whenever energy is included, that step is irreversible. Okay. If there is no energy included, like this step from glucose 6-phosphate to fructose 6-phosphate, it can be reversible. So first we add phosphate to the glucose, then we change the glucose into fructose. It still has a fructose, and the enzyme is called isomerase because it is an isomer. Then one more phosphate is added to that fructose phosphate molecule, and it becomes fructose 1,6-bisphosphate. And these phosphates are coming from the ATPs. Okay. This fructose 1-bisphosphate then breaks down for the first time and makes 2 molecules. So from here onwards, you will see 2x everywhere. 2 molecules of a 3-carbon molecule, which is called triose phosphate. The name is glyceraldehyde 3-phosphate. And you have studied G3P before also. Remember G3P? Photosynthesis? Calvin pathway? Perfect. So this is where the G3P again comes here. So this G3P has 1 phosphate. Now there will be more phosphates added and it will become 1,3-bisphosphoglycerate. But remember, these are all 3-carbon molecules, so 2 twice. From this step onwards, when this 1,3-bisphosphoglycerate is formed, now the energy starts coming back. So you start gaining from the pathway. Remember, during the investment phase, we invested 2 ATPs. So during the payoff phase, you get 2 ATPs at this step, where 1,3-bisphosphoglycerate converts into 3-phosphoglycerate or 3-phosphoglyceric acid. So because there were 2 molecules, 1 ATP from each molecule comes out. So 2 ATPs are recovered. So the net benefit or net loss is 0 here. Now at the last step also, there is one more payoff of ATPs and 2 more ATPs are recovered, okay, along with water at the second last step. So we get 2 molecules. So in glycolysis, the net benefit is of 2 ATPs. Is it clear? So this is a question that can be asked, that per molecule of glucose, how many ATPs are produced? Okay. So if they're talking about the net benefit, then it is 2 molecules of glucose are obtained. Okay. But if they're asking about overall, how many molecules of ATP are invested and produced? So 2 are invested, 4 are produced. The net profit is 2. Okay. But 2 is very less an ATP coming from 1 molecule of glucose. So that's why prokaryotes, which are anaerobic organisms like bacteria, they have very less energy to rely upon through glycolysis, because their glycolysis is the only mode of energy formation. They cannot do oxidative breakdown because they do not utilize oxygen. At the end of glycolysis, you started with 1 molecule of glucose and you are left with 2 molecules of pyruvic acid or pyruvate. Now this pyruvate goes into your other cycles in the mitochondria that we will talk about later on in this chapter. Okay, probably today. But this was a mode of energy formation which takes place in every living organism on the planet. Okay, from bacteria to humans, and it gives us 2 ATPs per glucose molecule and does not. Yes, Hiram.
So it's an anaerobic process.
Yes, it's an anaerobic process because it does not require oxygen. Okay. Even if oxygen is present in the surrounding, even when organisms like human, when they do glycolysis, oxygen is not required. Even if it is present, you will not use it because there is no step at which you need to use oxygen. You understand? So glycolysis is the most primitive and conserved methods of producing energy inside the cell. And when bacteria evolved first, this pathway came up, and it is present in bacteria, yeast, fungi, plants, animals, insects, amphibians, reptiles, birds, everywhere. Okay. Regardless of the organism, whether it is aerobic or anaerobic. Okay, this process, and that's why we just get 2 ATPs benefited out of this pathway. Is it clear, Iram? Cool. So glycolysis, as I said, as I told you, does not uses oxygen. So in today's class, we will take up, there are other ways also. One more method of producing energy where oxygen is not utilized is called fermentation. You know about fermentation? I'll not say oxygen is not utilized. Sorry. So in glycolysis, oxygen was not utilized at all. In fermentation, there is what you say, incomplete oxidation of glucose is achieved. So write down first what is fermentation. Write down, in fermentation, the incomplete oxidation of glucose is achieved under oxygen deprived or anaerobic conditions. And just to tell you that fermentation is studied very well in yeasts, fungi. Okay. Second point is, what happens here is, remember that first thing that happens for glucose is glycolysis. After glycolysis, we are left with pyruvic acid, right? So in fermentation, when there is oxygen deprivation or less oxygen present, then what happens is this pyruvic acid or pyruvate is converted to CO2. But as I told you, not all the pyruvic acid can be oxidized. So pyruvic acid or pyruvate is converted to CO2 and ethanol. CO2 and ethanol. Now you know what is ethanol? Ethanol. Anyone knows the chemical formula for ethanol? Methanol, ethanol. You must be knowing all these functional groups, right? All means OH, right?
Yes, sir.
Yeah, ethanol, the formula. Anyone knows? How many carbon should be there in ethanol? Tell me.
2.
2, right? Okay, let's do some calculation. How many carbon should be there in pyruvic acid? Let's go up. See, pyruvic acid, it is a 3 carbon molecule. Can you see 3C molecule? Iram, Devangi? Yeah. We started with a 6 carbon molecule, which is glucose. In every cell, when glucose enters, the first pathway it enters into, regardless of what organism it is, whether it utilizes oxygen or does not utilize oxygen or whatever, the first pathway, the default pathway in which glucose enters is glycolysis, where oxygen is not required. Okay, so at the end of this pathway, you will be left with pyruvic acid or pyruvate, which is a 3 carbon molecule. Now, what you do to this 3 carbon molecule decides whether you are an aerobic organism or an anaerobic organism. So from here onwards, you can do a complete oxidative breakdown of pyruvate, or you can do a partial breakdown that happens in fermentation. Does it make sense, everyone? Yeah, Iram? So let's see a simple maths. Pyruvic acid is a 3 carbon molecule. CO2 is a 1 carbon molecule, right? And ethanol is a C2H5OH. Ethanol is a 2 carbon molecule. So 2 plus 1 is 3. You understand now why it is called an incomplete breakdown? Because this pyruvic acid, when you completely break down pyruvic acid, you make 3 CO2. Okay, so every carbon should be independent, like single. 3 carbon are here, so 3 CO2. And from 1 glucose, how many pyruvic acid come? At the end of
2.
glycolysis, sorry, come again, Devangi.
2.
2. So in glycolysis, from 1 glucose, 2 pyruvic acids are obtained. So how many CO2s will be obtained from 2 pyruvic acids? If you completely break it down under oxygen, full oxygen availability. Write down this 1 line rule. Okay. If you use full oxidative breakdown processes, write down, if full or complete oxidative breakdown takes place, if complete oxidative breakdown takes place. Then only CO2 will be produced. Then only CO2 will be produced as a carbon output molecule. As a carbon output molecule. So no ethanol, no pyruvic acid, nothing else, just CO2. And how many carbons do CO2 have? Hiram. See, if you people will not speak, then I will not know whether you have understood or not. And I will just move. Am I audible?
Yes.
Yeah. How many carbons does CO2 have?
One.
One. So if there is a complete oxidation of any any biomolecule which is carbon based, then at the end, whatever is the total number of carbon present in that biomolecule, that equivalent CO2 molecules will be produced. Because oxygen we are giving from outside, carbon is already present in that molecule. So we started with one molecule of glucose, okay, which is C6H12O6. At the end of glycolysis, we are left with two molecules of pyruvate. And pyruvate is a 3-carbon molecule. That's why from a 6-carbon molecule, we broke it down into two molecules of 3 carbon each. Now, if you completely break it down, if there is lots of oxygen available, so if there is an oxidative breakdown, then what should happen? 3 to the 6. So 6 CO2 will be produced, right? You understand? Everyone? Yes, no, something. Yeah. So this is exactly what this formula says. C6H12O6 plus oxygen you give, you get 6 CO2. Now you understand how you get 6 CO2? Because full oxidation happens. But now in fermentation, this is not possible. In fermentation, what happens is, there is oxygen deprived condition or anaerobic condition. So one CO2 is produced plus C2H5OH, ethanol is produced. Okay. So 2 plus 1, 3. Because it's coming from one pyruvate. So there will be 2 CO2 and 2 ethanol. 2 to the 4 plus 2, 6. 6. Is it balanced? So from this step onward, you can write whether there is fermentation happening or there is complete oxidation happening. You can understand. Is it clear? Iram, Devangi. Good evening, Jania. Perfect. So this is what happens in fermentation. Okay. So let's see how it happens. Okay. So this is the overall reaction. Now let's start with glucose again. So we start with glucose. and we undergo glycolysis. At the end, we reach at pyruvic acid or pyruvate. You can write anything. Now, on this pyruvic acid, which is a 3-carbon molecule, there are two things that can happen. Either this pyruvic acid, under less oxygen availability, can become lactic acid. Okay. Or this pyruvic acid can become ethanol plus CO2. Okay. Now, ethanol is a 2-carbon molecule, C2H5OH. And lactic acid is a, how many carbons are there in lactic acid? Anyone? Anyone knows the formula for lactic acid? I'm sure you have studied this in your chemistry. Lactic acid is C3H6O3. Okay. Is it clear, everyone? If we make the structure of lactic acid, it will be C with 3 hydrogen here, CH3, C double bond O, sorry, 3 carbons, right? CH3, CH, and then C double bond O, OH. So 1, 2, 3. C, or here it will be OH again. So this is the formula. This is the structure. Now it is a 3-carbon molecule. Ethanol is a 2-carbon molecule. It is simple. C, so H, and then OH, and then H and H. CO2, you know, it's simple. Now pay attention here. I will use a highlighter to show you things. Now see, if we start from glucose, okay, and if the pathway goes through glycolysis, makes pyruvic acid, and then this pyruvic acid gets some oxygen, right? Some oxygen is there to make CO2, because to make CO2, at least you have to break down this 3-carbon molecule partially, right? Yes or no? To break it partially, you need some oxygen. So if some oxygen is present, it will go this way, and it will make ethanol and CO2. Is it clear? But if there is no oxygen at all, and you are very, very deprived of oxygen, then this pyruvic acid will channelize itself into this pathway, and it will not break down this 3-carbon molecule into any further, and the 3-carbon molecule will just change from pyruvic acid into a lactic acid, but still there is an acid. So this arm is followed when very less oxygen is available. Okay, it is called partial breakdown, partial or incomplete oxidation, because at least some CO2 is formed. Do you see any CO2 forming in this pathway, in lactic acid pathway? Do you see that? No. So no oxidation happens here. Is it clear, everyone?
Yes, sir.
So when we go from glucose to ethanol and CO2, this is called fermentation. The other one is called anaerobic respiration, and it leads to lactic acid formation. This happens in our muscles when we are doing very, very strenuous exercise, or when we are pushing our muscles to the limit, or when we are like playing a lot, even for hours and hours, or we are running constantly. So our muscles get fatigued and they do not. So the rate of energy utilization by the muscles is more than what we are breathing in. So oxygen requirement is more in the muscles, and we are not able to supply muscles with that much amount of oxygen through blood flow. So muscles, in the absence or in the scarcity of oxygen, start forming lactic acid. They cannot break down pyruvic acid or the pyruvate any further. And this lactic acid gets deposited in our muscles. And this is where, after a very strenuous gym session or exercise or playing, you get pain in your muscles. Anyone has witnessed that ever?
Yes, sir.
Yeah. So now you understand what biology is happening in your body.
Yeah.
Yep. So this is the fermentation major pathways of anaerobic respiration. So write down, these are the
So
two.
you basically like get cramps in the muscles, right?
Yes, that cramp is because of lactic acid deposited in the muscle, and because it is an acid, it will cause pain. Okay. When a bee stings you or an ant stings you, it releases formic acid inside the body, in the muscles, and it pains, right? There is swelling and there is pain. When someone, as a kid, if we used to squeeze orange peels in each other's eyes, I was kind of a very notorious kid. So any one of you have ever, it happens sometimes when we are peeling the orange, something from it comes into your eyes, right?
Yeah.
So it burns. Why? Because orange peels have high concentration of citric acid. Do you understand? Lemon juice, if it goes in the eyes, it burns. Yeah. Or if you have a small cut in the finger and you touch a lemon juice or orange juice or something with it, it will burn a little. So lactic acid, formic acid, citric acid, these are all organic acids. So the accumulation of lactic acid in the muscles causes the muscle to feel pain, and you call it muscle cramps. Okay. So these two, this pathway from glucose to lactic acid 1 and glucose to ethanol 2, are the two major pathways of anaerobic respiration. Okay. So organisms which show anaerobic respiration, like yeast undergoes this fermentation. Okay. So from glucose to ethanol is fermentation. The yellow, what is marked in yellow, is a fermentation pathway. So that's why yeast is used in making alcoholic beverages, right? Is it clear, everyone? Okay, perfect. Okay, cool. So and also with the red I'm showing you, so in both these processes, what happens is, here NADH plus H plus molecule is used, and NAD plus is produced. So hydrogen is used, because you see that there are many hydrogen which are added. Okay, it comes from this. Here also the same thing. NADH plus hydrogen ions come in and go as NAD plus. So you see hydrogens added, right? It's important. So this is the full. Please draw it and let me know if there is any doubt. And the enzyme, I'm writing the enzyme name is green. Inside our muscles, when pyruvic acid is reduced to lactic acid, the enzyme which is used here, so you can see that hydrogen is being added. So the enzyme will be called lactate dehydrogenase. Lactate dehydrogenase, because it is okay. Lactate dehydrogenase. And the reducing, these two are the reducing agent is NADH plus H plus. So this is the reducing agent. Yes, so Devangi has a question. So our cells can never undergo incomplete oxidation. Yes, because we are aerobic organisms. Human cells should completely oxidize glucose in the presence of oxygen and should only produce CO2. So our metabolic waste is CO2. Sorry, our respiratory waste is CO2. Not lactic acid, not ethanol. Okay, we don't make ethanol in our cells. We don't make lactic acids in our cell. But sometime when we are doing strenuous exercise and our muscles are inadequate, our muscles get an inadequate supply of oxygen, then our cellular respiration pathways might shift. And because oxygen is not available, so the enzyme lactate dehydrogenase may cause the pyruvic acid to become lactic acid. And this lactic acid remains stored in our muscles for some time and causes pain. Over time, this lactic acid is, again, when there is again oxygen available, when you will sit down for one day or 2 days, you will feel that cramp, but you will be breathing normally in that time, drinking water, so you will get more oxygen, and you will again break down this lactic acid to CO2 molecules. Okay.
Yes, sir.
Okay. Very good. Is this clear, everyone? Should I move forward? So anaerobic respiration is clear, Hiram? Okay, perfect. So let's go forward. And now let's start with aerobic respiration. Till now, we just talked about anaerobic. So glycolysis is which has nothing to do with oxygen. So it is not dependent on oxygen. And anaerobic can happen in the absence of oxygen also. But aerobic respiration, how do we define aerobic respiration is because the only waste material that you will get out of aerobic respiration is your CO2. So complete full oxidative breakdown of substances. So write down aerobic respiration is the process. Aerobic respiration is the process that leads to complete oxidation. That leads to complete oxidation of organic substances like glucose, okay, in the presence of oxygen. In the presence of oxygen, to release CO2, water, and large amounts of energy. Okay. Is that clear? Now remember, we say glucose, but remember that aerobic respiration is not, it cannot happen without glycolysis. So the first step, regardless of what kind of organism you are, what kind of respiration you do, when glucose enter the cell, the first thing that happens is glycolysis. So after that, whether you are doing aerobic respiration or you are doing anaerobic respiration, that starts with pyruvic acid. In an anaerobic respiration, the pyruvic acid does not get enough oxygen to be broken down and either forms lactic acid in our muscles or forms ethanol plus CO2 in case of yeast. So this process is called fermentation. Is it clear? So in aerobic respiration also, whatever is the effect of oxygen, we will start seeing it on pyruvate onwards, because from glucose to pyruvate is the same. Is it clear? Iram, Jania, is it clear for all of you? Okay, Iram?
Yes, sir.
Yeah. Okay, so again, let's write down some points first. Let me dictate you things. Write down. Aerobic respiration takes place in the mitochondria. Where was glycolysis happening? Where was glycolysis happening, people? In the cytoplasm. Okay. Glycolysis was happening in the cytoplasm. That's why it happens in all organisms, even bacteria, because bacteria does not have mitochondria. Bacteria, for that matter, does not even have nucleus. So bacteria has no membrane bound organelle. It just have a cytoplasm. If glycolysis can happen in bacteria the same way it happens in human, it simply means that it is happening in something which is common in both, and that is cytoplasm. Okay. Yeah. But aerobic respiration happens in mitochondria. That's why no prokaryote can do aerobic mitochondria based aerobic respiration. Only eukaryotes can do that.
So,
Yes, Iram.
so in cell chapter, we learned that in bacteria there is an invagination, which is extension of plasma membrane called as mesosome, that is equivalent to mitochondria, and that does aerobic respiration.
That helps in aerobic, yes. So about mesosomes, it is when we say that something is equivalent, we just mean that so if you see, like, I think this is what you're talking about. So it has an outer membrane, and you will see that sometimes the inner membrane undergoes and forms this kind of invagination, right? And this is known as mesosome. So that's where you say that we have aerobic bacteria as well, right?
Yes.
Correct. So they enhance, they support respiration in a way where there are two things. So first thing is, most important function of mesosome is it helps in the replication of DNA, protein synthesis, and the synthesis of cell membrane itself. But because it is a big membrane inside a small membrane, it also helps in respiratory functions. So that is equivalent because it helps in aerobic respiration in a bacteria. Is it clear?
Yes.
So it has roles in aerobic respiration, but saying that, but it is still not as efficient as a what do you say, as a mitochondria, because we'll see what mitochondria does that mesosomes cannot do. Okay.
Yeah.
It is not a true organelle. Okay. So it takes place in the mitochondria, whereas glycolysis only takes place in the cytoplasm. Very well. But good point you brought in. Thank you, Hiram. Just one second. Am I audible? Yes. Second point, write down. In aerobic respiration, the final product of glycolysis, which is pyruvate. So in aerobic respiration, pyruvate is transported. And of course, it has to be transported, right? So pyruvate is transported from cytoplasm to mitochondria. Is it clear? And there are two things that is happening after that. So first is, in aerobic respiration, you have lots of oxygen available, and the cell can use that oxygen also. So the first thing that happens is the complete oxidation of pyruvate. Okay, where you remove all the hydrogen atoms one by one. Okay. So as I told you, from pyruvate, if you remove all the hydrogen atoms one by one, so if you don't know, how many of any one of you know the structure of pyruvate? How does pyruvate, like what is the structure of pyruvate? Anyone knows? Or pyruvic acid? No? So this molecule pyruvate, what is the formula? It's a 3-carbon molecule, but it is C3H4O3. Okay. And if you look at the structure, it is C, on one side it is OH, on other side double bond O, another C, double bond O, then another C, H, H, H. This is a pyruvate molecule. 1, 2, 3 carbon, 1, 2, 3, 4 hydrogen, and 1, 2, 3 oxygen. Okay. Now this, in a stepwise manner, all these hydrogens are removed. Okay, so when you remove hydrogen, it is called oxidation, right? Oxidation is either adding oxygen or removing hydrogen. Is it clear? Is it clear, everyone? Okay, very good. So just remember the structure. It is not given in NCERT, but just knowing the structure helps a lot. So first thing, write down. First thing that happens in aerobic respiration is a complete oxidation of pyruvate by removal of all the hydrogen atoms. That leads to formation of 3 molecules of CO2. Remove formation of 3 molecules of CO2. So this is the first thing that happens. So let me write this in green. First thing, all hydrogens removed from pyruvate. Okay. And this process is called complete oxidation. And 3 molecules of CO2 produced. And the second thing that happens, this is just the part of the process. And the second thing that happens is all the hydrogens that has been removed, they carry electrons, right? Hydrogen is the simplest atom carrying 1 proton and 1 electron, correct? Yeah. So all these, if you remove 1 hydrogen, you also have 1 electron with it. So write down the electrons removed as a part of the hydrogen. Electrons removed as a part of hydrogen is passed on to O2 molecule. Now this molecule that is present, this is what we breathe in. Okay. And when you add hydrogen to O2, what will be formed? Water, correct? So do you understand that CO2 also is formed? Hydrogen is also added to O2 to form water. And in this process, when you are doing, this is called reduction of oxygen. So when you reduce oxygen, you produce ATP. ATP is synthesized, and how it is done, I will teach you in detail. ATP is synthesized, and this is what we wanted to achieve. So through this second process, you are making additional ATPs apart from those 2 ATPs that were formed when glucose was broken down into 2 molecules of pyruvate in glycolysis. So in bacteria, because they cannot do this, these 1 and 2 that I have written, because that is to be done in mitochondria. So they cannot do this process. So they only have to survive or rely on the 2 ATPs produced during glycolysis. So their only source of ATP production is glycolysis. Is it clear, everyone? Thumbs up. But for organisms with mitochondria, you have now chance and you have now opportunity to produce more ATPs through a full aerobic respiration inside the mitochondria. Okay. And also, for the sake of your entrances, note down that this first step and the second step, they happen in 2 different locations. The first step is happening where you completely oxidize pyruvate by removing hydrogen atoms. This step takes place in the mitochondrial matrix, which means inside the mitochondria. Mitochondrial matrix. It was asked in your entrance. Whereas the electrons which are removed as a part of hydrogen, this passing on of electron to oxygen, this happens in the inner membrane of mitochondria. Inner mitochondrial membrane. Is it clear?
So,
Yes, Iram.
so adding hydrogen to O2 molecule will give you water, right?
Yes.
But we require 2 molecules of hydrogen, right, to form H2O.
Yes, right. So for 2 molecules of oxygen, we need 4 molecules of hydrogen when it is added.
Okay.
And we have lots of hydrogen in the cell. We'll talk about it. Okay. So here I'm just laying the foundation for you so that this very complicated topic becomes very simple in your head. So you all till now, let me give a quick recap in 30 seconds. Till now, what we have studied, we are going very, very molecularly deep. So it's very difficult to keep everything, remember and memorize everything. So let's go like a flow. Till now, we realize that everything starts with glucose, correct? We eat food, we get glucose out of it. Whether you are eating a biscuit or a Karela does not matter. What you are getting for energy is glucose out of it. Is it clear? You all know Karela, right? A bitter gourd. Yeah? Yes, no?
Yes.
Correct. So it all starts with glucose. Glucose goes inside each and every cell. And we know for glucose to go inside the cell, we need insulin signaling. Correct? This we have already studied, just connecting some dots. The moment glucose goes inside the cell, glycolysis starts. Okay? Which has nothing to do with oxygen. Till now, this is a common universal process happening in every organism, bacteria, human, anything which is living. So glucose in the presence through the process of glycolysis, through many enzymes that come one after the other, and exactly 9 steps that are there in the process. To do glycolysis, the cell first invests 2 ATPs and in the investment phase, and then gets 4 ATPs back in the payoff phase, and there is a net profit of 2 ATPs from every molecule. And those 2 ATPs can be used for other processes to maintain the life, because there are millions of glucose molecules broken down every minute in a cell. So there will be millions of ATPs produced, twice the millions. Okay, cool. Till here, okay. Bacteria can't move ahead of this most of the times, except for some bacteria that has mesosomes that Iram brought about. So I'm not going to include that in this chapter because that will include make it a little complex. So we are keeping mesosomes and the bacteria that are capable of doing something similar to mitochondrial respiration away. Okay, Iram, just for the sake of simplicity. We can discuss this later on once we are done with all these things. Is it okay, Iram?
Yes, sir.
Yeah. So till now, just know that bacteria cannot move ahead of glycolysis. So they stop there. Now, from there onwards, there are pyruvate is left. We got pyruvate. Now, what to do with pyruvate? Bacteria can't do anything with pyruvate. So they will either, throw it out or use it somewhere else, but not to produce energy. Done. But yeast can go one step further, or our muscles under a oxygen deprived condition can go one step further to either partially partially break it down, like it happens in yeast, where you break it down into CO2 and ethanol, or to convert it, reduce it into lactic acid, which can later on be again, broken down when oxygen is available. So that is called anaerobic part of respiration. Then we come to aerobic part. Now here we can exploit the full potential of pyruvate because we have lots of oxygen and we also have mitochondria. So what we can do here is, in the mitochondrial matrix, we can, strip apart this pyruvate, remove all the hydrogen, make it a bare minimum, and naked CO2 molecules we can make. Okay, this is happening in mitochondrial matrix. A simultaneous process is happening where the hydrogen that we are removing from pyruvate and making CO2, we are passing that hydrogen on to oxygen. And this is somehow helping us to make more ATPs. So in aerobic pathway, you have now the chance to make more ATPs apart from those 2 ATPs that you made in glycolysis. Till here, everything is clear to everyone. We are all on the same page. Thumbs up. Only I can see Devangi's thumbs up. Jani has also clear. Hiram? Cool. Now this part where electrons are passed on is called electron transport chain. Electron transport chain is a characteristic feature of aerobic respiration. And through electron transport chain, we produce lots and lots of energy. Okay. So the foundation is set for all of you. Let's move. Okay, now write down. The reaction, the biochemical reaction of aerobic respiration through pyruvate dehydrogenase. Just like here it was lactate dehydrogenase, in the mitochondria we have pyruvate dehydrogenase or pyruvic dehydrogenase. Anything you say, the same thing. Now this is the reaction. So we have pyruvic acid. I am writing the names. In exam you can also write the name. If you forget the chemical formula or structure, not at all a problem. But I'm writing both, let's say, for your sake. C3H4O3, right? So pyruvic acid enters mitochondria. The first thing that comes and interacts with pyruvic acid is coenzyme A. It's called CoA, also known as coenzyme A. Write down. And comes your NAD plus. Okay. Remember, this is going to take the hydrogen away from pyruvic acid and it will add CoA to the pyruvic acid. So what is going to happen here is, the hydrogen which will leave from here will go to this, and when the space will be empty, this CoA will go to this. Is it clear? So as a result, what happens is the enzyme pyruvate dehydrogenase, and for this enzyme there is a cofactor which is magnesium. Magnesium is a cofactor for many enzymes in our body, including especially many respiratory enzymes and many photosynthetic enzymes. So for pyruvate dehydrogenase, this Mg2 plus is a cofactor. Cofactors are like keys to the car. So what you drive is the car. So the engine, the car, everything is very important. But can you switch on a car without the keys? Yes, you can in a theft mode, but let's keep things simple. Can we switch on a car without a key?
No.
No, right? So Mg2 plus is a cofactor that activates this enzyme and allows it to do what it does. Okay, so in the presence of this enzyme, what happens is that pyruvic acid plus coenzyme A becomes acetyl coenzyme A. Acetyl CoA. So pyruvic acid plus CoA becomes acetyl CoA. And what is removed? All the carbon is removed and it becomes CO2. Plus all the hydrogen that was removed from pyruvic acid that went to NAD plus, it will form NADH plus H plus. This H plus now, if you can see this in one go, let me make some notes. So as I told, everyone has written this reaction completely. So this is the complete reaction. Now what happens? From pyruvic acid, we are removing hydrogen. We are adding CoA to the pyruvic acid. So this hydrogen is here, and some hydrogen goes to NAD+. Okay, and this becomes NADH plus H+, and this CoA, when it's bind to pyruvic acid, acetyl CoA, it becomes acetyl CoA. So one carbon retains. So to just explain you what do we mean by acetyl CoA, so sorry, acetyl CoA is a 2 carbon molecule. Okay, so one carbon removes, and from pyruvic acid, one carbon becomes CO2. And anyways, we have no shortage of oxygen now because we have lots of oxygen in pyruvic acid itself. So remember, in pyruvic acid only we have 3 oxygens. So 2 oxygen along with the carbon comes out of pyruvic acid and becomes CO2, and 2 carbons stays and become acetyl CoA. Okay, hydrogen comes out, reduces NAD+ and also is present as H+. This H+ will go on to reduce O2, which we breathe in. Remember, in the mitochondrial membrane, the O2 is reduced by the hydrogen. Is it clear, everyone? So these arrows you don't have to draw. This was just to explain you the chemistry. Everyone understood what's happening here? Perfect. So write down this reaction, and this is the reaction for what happens in the mitochondria. Anyone has any doubts still here? Any doubts? Okay, so write down a few important points. During this process, this above process, so where you have written this reaction, just write down, during this process, of pyruvic acid becoming acetyl CoA, and remember, this is all happening in the mitochondrial matrix. So during this process where pyruvic acid becomes acetyl CoA, 2 molecules of NADH are produced. So if you balance this reaction, you will realize there were 2 pyruvic acids to begin with, right? Because from each glucose, 2 pyruvic acids come. Correct? So you will need 2 coenzyme A to make 2 acetyl CoA, and 2 NADH will be produced. So everything will be 2. Do you appreciate, people, why everything will be 2? Perfect. So 2 molecules of NADH are produced. And this is an important molecule. We will realize it later why it is important, probably in the next class, but 2 molecules of NADH are produced. And why is the cell doing all of this? Because till now we have not produced any energy. And in the mitochondrial matrix, we do not produce any energy. So the energy is produced in the inner membrane, right? ATP is synthesized in the inner mitochondrial membrane. And what is happening in the inner mitochondrial membrane? Electron transport chain, to which we will come later in the next class. But let me tell you, this whole mumble jumble circus was being done so that we could make this molecule. Acetyl-CoA is a very important molecule because this molecule enters there's a pathway called Krebs cycle. Okay, it is also known as TCA, tricarboxylic acid cycle. So we will start with TCA in the next class, but write down the acetyl-CoA which is produced. Now remember how many acetyl-CoA were produced from one pyruvic acid? How many acetyl-CoA will be produced from one pyruvic acid? Devangi, Iram, Jania, quickly.
1.
Perfect. I was just trying to trick you. So how many acetyl-CoA molecules will be produced from one glucose?
2.
2. Often students get confused because acetyl-CoA is a 2-carbon molecule, glucose is a 6-carbon molecule. So sometimes what they in entrances what I have seen is they write the question like this: how many acetyl-CoA, which is a 2-carbon molecule, will be produced from a glucose, which is a 6-carbon molecule, in a aerobic respiration? So just to trick you, okay? So you will say 3, but remember 2 because 2 CO2s are also formed. Okay, is it clear, everyone? Devangi, Iram, and Jania? Yes, so write down the acetyl-CoA which is produced enters the tricarboxylic acid cycle, the TCA, tricarboxylic acid cycle, TCA cycle. It's also called as TCA cycle. Also known as Krebs cycle. Krebs was the name of the scientist, Hans Krebs, who first, figured it out, this cycle. And after TCA, we will talk about electron transport chain. So the last 2 topics of this chapter, TCA and electron transport chain, are the places or are the reasons or are the cycle where energy will be produced, lots and lots of energy. And to just give you context, how many ATPs we were able to form from glycolysis per glucose? How many ATPs we get per glucose from glycolysis?
2.
2 ATP, correct? Do you understand? You know how many total ATPs per glucose we can produce if we do a complete oxidative respiration? Any idea? Inside the mitochondria, when the acetyl-CoA enters, pyruvic acid enters and become acetyl-CoA, from this molecule onwards, now you will understand the value and importance of this molecule. We never realize in normal textbooks how important is acetyl-CoA. Glucose, everything started with glucose, of course. Glucose is the dadaji of this whole, this whole Khandan network. So he's the most revered, it is the most revered or respected. But the point is, glucose through glycolysis just gives 2 ATPs. But this acetyl-CoA in the mitochondria through this pathway gives 36 ATPs per cycle. Do you understand that? So total, you get 38 ATPs at the end of oxidative respiration. Do you understand? Isn't it amazing? So all of a sudden, a cell with a mitochondria from 2 can get 38 ATPs, 36 to 38. Amazing, right? This is the reason for multicellular life to be the way it is. Everyone understands that? Iram, Devangi, and Jania. So in the next class, what we will be doing is we will start with this TCA cycle, okay, in the cell, and we will understand how this TCA cycle, which is also known as tricarboxylic acid cycle or citric acid cycle. Why it is called citric acid? Because tricarboxylic, the first, you remember how we used to name the cycles? Do you remember oxaloacetic acid cycle, OAA pathway, or TGA pathway, triglyceric acid, right? We always name these cycles with the first stable product that they make. So the first stable product in this TCA cycle is citric acid. Yes, the same citric acid which is present in your oranges and lemon. Okay, so it is also called the citric acid cycle. So 3 names: TCA, tricarboxylic acid cycle; Krebs cycle, name of the discoverer; or citric acid cycle, which is the first stable product in the pathway. Is it clear, everyone? All 3 are the names of the same pathway, and we will talk about it in the next class, and we'll try to finish this chapter.
So,
yes, Iram.
So I had a question to discuss that came in my exam.
Yes, please.
It says that, 1 minute. Why is insulin not given orally, but it is injected into the body?
It's very simple. What is insulin?
Insulin, it is
a protein.
It's a protein.
So the moment you will feed any organism a protein, that protein will go into the stomach. In the stomach, we have pepsin. Pepsin is a protease. It will break down the protein into amino acids. Insulin is gone. It's no more insulin. It is just amino acids.
So it's because of the digestive enzymes.
Yes, it is because of the proteases that we have in our stomach and in our intestines. So we need insulin to reach the blood in the form of insulin. Okay, so that's why any protein that we want to reach inside the blood in the form of the same protein where it is functional, so insulin is injected in the blood. If you feed it orally, it will be degraded.
Okay.
Okay.
Yeah.
Simple. Make sense?
Yes.
You know, that's why venom, if you have no wound in your mouth, no wound, which is like a very rare, we always have some or the other small wound or an open blood vessel. But if someone has no wound in their mouth, they can actually drink snake venom in small amounts, and that will not do anything. You know why? Because snake venom is again protein. The moment it goes in our stomach, it gets digested.